Hello and welcome to Nikolai's genetics lessons. In this video I'm going to talk about Dihybrid cross, and here's a problem. Mendel crossed purebred wrinkled green seeded plants with purebred round yellow seeded plants. Round seeds are recessive to wrinkled and yellow seeds are recessive to green. The F1 progeny were self-crossed. Which of the following numbers are most likely results from this cross? And here's the 5 answers to choose from. If you feel that you can solve this problem, you may stop video here, try to solve this problem on your own first, and when you would be ready you can run video again and you can compare your answer with my answer and explanation. In order to solve this problem, we have first list genotypes of both parents. So we have two parents, parent 1 and parent 2, and we know that first one is wrinkled with green seeded with green seeds. So also we know that round seeds are recessive to wrinkled. We also know that wrinkled can be made by two dominant alleles, and another variant would be one dominant allele, another is recessive allele, but phenotypically they would look the same, wrinkled. So which one genotype to choose? And this word would help us. So we have purebred here, and that means that genotype would be homozygous. So if we would use capital R in order to designate this allele, we would have for parent 1 two capital R alleles for this locus. And we also know that it is green seeded, and here also we have information that yellow seeds are recessive to green. So green color of the seeds would be dominant. Once again, because this is pure breed, the genotype would be capital G, capital G. So this is genotype of the parent 1, and parent 2 is also pure breed, and it is round. And round phenotype only can be made by one genotype, that is small r, because if we would have capital R, this would be wrinkled, and if we would have heterozygous, this wouldn't be pure breed then, and if we have two recessive alleles, this means pure breed round. So next trait would be yellow seeded, and that means that yellow color is also recessive to green, and it is pure breed. So this is going to be small g genotype for the color. So this parent would be wrinkled and with green seeds, and this would be round with yellow seeds. So now we have genotypes of the parents, and this is going to be parental generation. So this line stands for the generation of the parents and their genotypes. We have to cross them in order to get F1 generation or filial 1 generation. And as you see, for the first gene, parent 1 only can produce one type of alleles, that is capital R, and parent 2 only can produce small r alleles. So we know that in F1 generation, genotype for the this locus would be capital R and small r. So capital R comes from this parent, and this can be any, this capital R or this capital R, it doesn't matter. And the same rule applies to the parent 2. Parent 2 only can produce small r alleles. So genotype of the F1 generation for this locus would be capital R, small r. And as for the second locus, parent 1 only can give capital G, capital allele or dominant allele. So this is going to be capital G, and parent 2 only can give to the progeny only one of these recessive alleles. It doesn't matter which one, but only one type, that is recessive and small g. So phenotypically, F1 generation would be wrinkled and also would be green. So phenotypically would be the same as parent 1, but genotypically would be different. So now we are told that F1 progeny were self-crossed. What does it mean? And that means that we have to cross this genotype with itself. In other words, it's also called selfing. So self-pollination, for example, if it is plants, or self-crossing if it is animals, for example. So what is the genotypes would be of the F2 generation, and this time we cannot list all the possible genotypes in one line because there are going to be many different genotypes because alleles and traits would segregate. So now let's think what kind of gametes each parent can produce. For example, this parent can produce gametes are haploid, so only one allele out of each allelic pair we can choose. For example, within this couple we choose capital R allele, and within this couple we choose capital G allele. So let's list this genotype, that is capital R and capital G. The second variant of the gametes would be capital R and small g, so capital R and small g. Third variant would be small r and capital G, and this is going to be small r capital G. And the last variant would be small r and small g alleles. So this is small r and small g. And as you see, the genotype of both parents are the same. So that means that in order to find all the possible variants, we have to cross genotypes of these two parents, or in other words, we have to build a Punnett square. So if we have all the listed genotypes for parent 1 on top, for parent 2 we put the same genotypes on the side. So capital R and capital G, capital R and small g, small r and capital G, and small r and small g genotype. And now we just built a Punnett square, and as you see we would have 4 rows and 4 columns, and total number of the cells would be 16. But that doesn't mean that we are going to get 16 different genotypes or 16 different phenotypes. So for example, here we would have one dominant allele from one parent and another dominant R allele from the second parent. So genotype would be capital R, and also we would have one dominant allele G from one parent and one dominant allele G from the second parent. So capital G genotype. Here we would have capital R and capital R, and capital G, small g, capital R, small r, and capital G, capital G, capital R, small r, and capital G, small g. Next column, capital R, capital G, small g, capital R, small g, capital R, small r, and capital G, small g, capital R, small r, and small g. So we have done. Next column, capital R, small r, and capital G, capital R, small r, and capital G, small g. Small r here, and capital G here. Small r here, and capital G, small g here. capital R and capital R, and small r here, and capital G small g here, capital R small r, and small g, and here we have small r, and capital G, and small r, and small g here. So we are done, and with the next step we have to analyze what kind of phenotypes we have here. So we have all the listed genotypes, and now we have to find what is the phenotypes we got here and what is the ratio. So here in the first cell we have capital R, that stands for the wrinklet, and capital G stands for the green. So this is going to be wrinklet and green. Next cell we have capital R. Also this means that this is going to be wrinklet, and we have capital G and small g. That means it's also going to be green. So wrinklet and green. Next we have the same phenotype, because all the heterozygous would be wrinklet for the first allelic pair, and wrinklet and green also we would have here. So next column we have the same phenotype here, but next cell, as you see, we have wrinklet, but we have homozygous recessive for the green color, that means yellow color. So wrinklet and yellow. Next we have once again wrinklet and green, and next we have wrinklet and yellow. So wrinklet and yellow. Here we have once again wrinklet and green, here, and this time we have round and green. So round and green, round and green here, and wrinklet and green here, and as you see here we have wrinklet and yellow, and round and green here, and round and yellow in the last cell. So what is our ratios? Let's count. So first phenotype would be wrinklet and green. So we have 1, 2, 3, 4, 5, 6, 7, 8, and 9. So we have 9, that is wrinklet and green. The other phenotype would be wrinklet and yellow. We have 1, 2, and 3. So 3, wrinklet and yellow. The other phenotype would be green and round. We have 1, 2, and 3. So going to be green and round. And the last phenotype, as you see here, would be yellow and round. So we have only one such phenotype here in our table, yellow and round. So as you see, our ratio would be 9 to 3 to 3 to 1. Now let's return to given answers that we have to choose from, and let's check what the ratios we have here. And answer A, we have ratios that is 1 to 1 to 1 to 1. So this is wrong answer. And for example, answer E, we have ratio as 3:1, so also we can cross out this answer. Answer D, we have ratios as you see, if we divide by 10, for example, this is going to be 6 to roughly 2.5, 2.5, 2.5. So we also can cross out this answer. And let's analyze answer C. We have 1 to say 2.5 or 3, 2.5 or 3 here, and 9. So it looks like it can be our answer. But answer B, we also have ratio that is 9 to 3 to 3 to 1. So which one to choose? And let's then analyze phenotypes. So here we have 9 that is wrinkled green, and here we have 9 that is round and yellow. So ratios here and here are the same, but phenotypes are different. And as you remember from our table, we got 9 ratio that were wrinkled and green and not yellow and round as you see here. So we can cross out this answer. Those from the first glance, it was like most probable answer, because ratio here were 9 to 3 to 3 to 1. But in answer C, we have different order. We have 1 to 3 to 3 to 9. But actually order doesn't matter. What is matters is phenotypic ratios. So as you see, our correct answer would be C. And this is ratios. And ratios are the same that we got here. Exactly the same as we got here. So this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.